I could just say, screw it all, put it on GitHub and make people compile themselves. This is JavaScript and Python, so it doesn't matter. It's just system agnostic. That's why I'm making it this way. I'm trying to make some fancy installer for some non-computer genetic person to be able to use my browser.
Because we have a meeting schedule, and then the meeting schedule right it's like 5:30 and everything.
Understand that. Yeah, I do have.
Oh, okay. I feel like whatever.
Dan Burns wanting some
Like a lot of the side dishes.
software installed. Made a bit of the old state.
Even ours, like rice.
Hidden Markov model, SAM. And it's like it wanted all kinds of libraries and goes, well, I can make tons of symbolic links all over your computer because it's looking in user live.
Finding a letter of support commission for this budget.
And I'm looking at it like, I'm doing all this, but I'm thinking, I am just destroying my computer right now. If I do a defrag, it's not going to know what to do.
You don't have to
It's
write
connected
anything,
here,
just
but
tell me
it's
to
connected
check
here, but it's updating from this. We're gonna
it. I want to take care of it. This is-
The problem is, like, I try something and it works. It's like, I'm not taking it.
Your text is where?
So if I need to replicate this later.
I know that
You haven't written it. I see.
this is the audit, answer the audit form. And for the Amy. It's not really fast, ultra fast.
Alright, welcome everyone. Tools and Technology Seminar Series. Just as a reminder, of course, this seminar series is a little different than your typical research talks, just in that it really focuses on tools, technologies, methodologies, either developed here or just being used by researchers here. There's a sign-in sheet going around. I know many of you have already signed it, but if you haven't, please sign it, and if you guys can continue moving it along, that would be appreciated. Thank you. So today's speaker is Santiago Schnell. He is the John A. Jacquet Collegiate Professor of Physiology, Interim Chair of Department of Molecular and Integrative Physiology, and Professor of DCMB.
Well, thank you for the invitation. So I think we have this invitation over a year ago, and the person who was supposed to be talking was Mike Vincent. But you know, he was a research associate in my group, and he ended up getting a position in Northwestern as a graduate student, and then he left, which broke my heart because he did a fantastic amount of work in my group, particularly on this area of protein informatics. And, you know, let me just tell you. So I'm not entirely sure how many of you are familiar with my work, so I should probably emphasize what I do. So I wear two hats as a researcher. So my first hat, as a single investigator, the focus of my group is on reaction kinetics. So we specialize on modeling complex biochemical reactions with the goal of first developing methodologies for understanding the dynamics of the reactions in time and space, and secondly, for developing tools to measure effectively the rates of reactions. So as a single investigator, my lab is a measurement lab. So we're focusing on how effectively to measure biochemical reactions and standardized approaches which are important for biochemistry. Then my second hat is as a science team member. And as a team scientist, what I do is that anything that boils down to biochemistry that I think I can model, and it would help you to sort out a physiological or biological question, we then help you to put a mathematical and computational model to solve it. And we don't limit ourselves to differential equation models, but we use essentially most of the mathematical and computational techniques available. Most of the work focus on computing and mathematics, not on omics inclined work and data analysis, but we do a little bit of that. And well, the reason I wanted to talk about protein disorder is for a number of years we have been thinking about protein disorder. First, because we find that the definition of what a protein disorder is very lax. But at the same time, there are questions of whether protein disorder has a function from the biochemical and the physiological point of view. And you know, I have a strong view of whether protein disorder has a specific function or it helps to promote other functions, the change in the structure of the cell. So and let me tell you why I think this is important. So you probably have heard about the reproducibility crisis. And the reproducibility crisis goes across the board, not only because people are employing incorrect statistical approaches, but because as well we have undefined standards of what it means to use a term or to do a calculation. And in my opinion, this is happening with protein disorder. So when you talk to a scientist, every scientist would have a unique definition of what they mean by an intrinsically disordered domain or an intrinsically disordered protein. And I'm going to give you an overview of what is currently known at the moment and where we think we are making a contribution. And you know, I'm going to leave you to judge that. And well, one of the things that people have been doing in recent years is that there are international efforts to actually coordinate how to report effectively anything on the biomedical and the clinical sciences. And in my case, I have been heavily involved on the biochemical sciences on this international commission that is called the STRENDA Commission that is handled by the Bellstein Institute, where STRENDA stands by Standard for Reporting Enzymology Data. And again, as a single investigator, I told you that most of my work focused on measuring reactions. Well, 90% of the work we have done is on measuring enzyme catalyzed reactions. And I have been part of this commission where we're coming with guidelines of how to report the enzymology data. And we have developed a database where people can upload their experiments and then check if you put all the experimental protocols in there, whether people can reproduce those experiments effectively. And you know, currently the standard guidelines only focus on the experimental essays. The next step that we want to move forward is on the measurement protocols of how people are estimating the parameters. And well, my goal is that I wanted to do something with protein disorder of similar nature. Can I sit down the people who work on protein disorder and come with guidelines of what it means? You know, my results were in vain. So I started to work very hard, contact people who work on protein disorder, and you know, everyone couldn't agree whether we should be meeting. And those that agree on meeting, they say, well, it's my protocol that is the right one and not yours. So what I decided is split from those people and then come with my guidelines, and I'm going to explain you how they operate. But before that, I want to give a brief introduction of what protein disorder is. So currently what we know is that, you know, proteins generally they fall into a three-dimensional configuration from the amino acid sequence, where they are naturally following the trajectories, and those trajectories start to change the configuration of the protein until it reaches a low energy state, the lowest energy state, where is the native structure. And well, technically, when a protein is intrinsically disordered, you have reached that lower energy state. You have fallen to a lower energy state, but you cannot see a unique configuration. Let me show you what the general concept is. So people define intrinsic disorder, intrinsically unstructured disordered proteins. They don't have a single well-defined ternary structure in their native functional state. So they have a function. They have a low energy state. They seem to be working under physiological conditions, but a region of that protein or many regions of that protein, they cannot be crystallized. That's the way they're defined. So if you don't have a crystal structure, then you believe that you have an intrinsically disordered protein. And the reason we know this is because the proteins they can crystallize just small domains, and they can crystallize in complex because those disordered domains acquire configurations where they're in complex. And it has been observed that when you have that intrinsically disordered protein and binds with different partners, the disordered domain acquires different configurations. But those configurations are unique to the pattern, to the partner that they have in crime when they are doing the biochemical interaction. And you can have a locally disordered protein or a fully disordered protein, and the difference is that you have a small domain in one area, or then you can have multiple domains that are linked by other ones, and this would have larger regions. So let me show you what they look like. So if you have an intrinsically disordered domain, you will go to a low energy state, and in the low energy state, yes.
I think you have two concepts. One is free energy.
Yes.
One is energy.
Mm-hmm.
So native state is the lowest free energy.
State, yes.
But why you always say it's the lowest energy?
Well, it's just shorter, so I apologize.
But that's important because intrinsical disorder, the entropy you cannot.
Yes, well, you're absolutely right. So, and I appreciate that clarification. So well, let me just then come in here. So when you are in this state in here, in the free energy state, so you can have multiple configurations of the same protein, and effectively what is changing between the states is just the disorder domain is acquiring slightly different configurations, but the overall structure, the crystallized structure that you can observe in the protein data banks is essentially identical. The conformation is just varying in the free lowest energy state when you observe small changes in the disorder domains. Then we can have another case where you have just a small region that actually can be fully crystallized and have a three-dimensional structure, and then you have multiple regions that are highly disordered, and they could have different forms and shapes. So you can have more than 50% of the protein that is highly disordered, or you can have somewhere around half of the protein disorder. And the challenge is that when you talk with protein of what they do consider an intrinsically disordered protein, they will tell you that there are multiple definitions. In the literature, the state of the art, what is considered to be a highly disordered protein, they say that if you have a stretch of 30 consecutive residues that by any algorithm that would give you that they are disordered, then you say that you have an intrinsically disordered protein or an intrinsically disordered domain. So,
however big the protein is.
However big the protein is. So that's what is considered the state of the art. So if you go to the literature, people immediately start to call their protein intrinsically disordered if you observe 30 continuous residues. And I'm going to tell you the history of that. That's where it's coming from. That's coming from the Dunker group, which is a very well-known group on protein informatics. And they carry out this study where they investigated the intrinsically disordered in cell signaling and cancer-associated proteins. And effectively what they did is that they took 4 groups of proteins. So they took the proteins where they have human cancer-associated proteins, they have signaling proteins, then they have eukaryote proteins from the Swiss probe, and then they took the non-homologous protein segments that are well-defined with three-dimensional structures. And they calculated with their algorithms. So the Dunker group has developed multiple algorithms for estimating protein disorder, how the intrinsically disorder of those groups appears. Peter Dunker has the philosophy that signaling proteins, they tend to be highly disordered because the flexibility of the disorder allows the signaling proteins to act more effectively. And he claims as well that many diseases are the result that you have a large number of disordered proteins. So he would claim that cancer, diabetes, Alzheimer's, they tend to be caused because you have a high number of disordered proteins. So whether that's true, I think it's a questionable statement. But well, because it's
It's a negative argument there, it looks like, in the middle of the paragraph. It says that those that are transmembrane proteins, which have to be highly constrained,
yes,
have less disorder than these. So these have excess disorder based upon a very peculiar comparison.
yes. So but let me show you the comparison he does where he draws the threshold, because this is the interesting, and you can see how the definition comes in. So they effectively, what they do is that they put the 4 protein data sets, which are in here. Then they calculate the average length, the median length, and then the maximum length. And one thing that they do, which is very awkward, is that effectively what they do from this group is they have any proteins that are shorter than 30 amino acid residues, they remove them from the system. And once they do that removal, then they calculate the overall disorder, which is what they do in here, and they see that the signaling proteins, which are the orange ones, and then the cancer proteins, they tend to be, if they're greater than 30 residues or equal, they tend to have more higher disorder. The overall percentage of proteins which are disordered, it tends to be higher than those that are the crystallized and then the generic eukaryote proteins. And he does this as increasing the number of disordered regions. And he finds out always when you're above 30, then you discover that you have highly disordered proteins. That's the way he comes with the definition. So, and you know, the point of the article, there is no discussion of why they didn't select the smaller proteins. They didn't did any sort of statistical analysis to determine the whole proteome, how the disorder compares, and effectively by setting that threshold, so they just go with the length, that if you have 30 or more continuous residues to be disordered, then you claim that the protein is going to be highly disordered. You know, we were very uncomfortable with the definition. Yes?
From crystallography data,
Yes.
not cryo-EM.
Mm-hmm.
These are from the old crystallography data.
Well, remember that...
2002 paper.
Yes. Yes.
Did
So,
cryo-EM change that?
well, it hasn't changed. So the challenge that once that definition was settled, nobody has called it into question until we just write this recent paper a year and a half ago, questioning the validity of that assumption. And you know, the challenge as well with intrinsically disorder is that, of course, it's very difficult to define because, you know, you can, the only way you know that a protein is intrinsically disordered is if you attempted to crystallize it multiple times and you can't. But there isn't a formal definition, and the definition changes from algorithm to algorithm.
I think they've also seen it in NMR, studies of proteins in solution. That some of them can take on multiple
Configurations.
configurations.
Thank you.
But you know, the other thing is interesting, and I'm leaving this blank slide because when we were working on this, we had a lot of difficulties publishing the guidelines because we have, in two occasions, we have the infamous third reviewer rejecting the paper on the following grounds. So we were told that, you know, an intrinsically disordered protein on an intrinsically disordered domain is called that way because if you believe as a scientist that that domain is important for its function, then you call it an intrinsically disordered protein. Which is, you know, it's the same way that if you believe that an alpha sheet, so, or, you know, any other structure of the protein is important, then you can call, well, let me call this an alpha protein, because, you know, the alpha helices here, they play an important role on the protein, which is ridiculous by definition. But, you know, people, they tend to be very sensitive to these issues, and, you know, there's a bit of people when they discover that the protein has some disordered domain, then they want to call it intrinsically disordered because it sounds hot to call something disordered. So that's the way we have perceived that works in the literature. So what we try to do is, you know, publish guidelines across proteome, which are proteome-based to characterize disorder. And, you know, our assumption was very naive. So I work with a lot of colleagues in the clinic. And, you know, how clinicians determine that somebody is obese or how somebody is tall. So how we do the determination that, you know, somebody has a head that is slightly smaller or, you know, hands which are slightly shorter. You know, we do it by population-based metrics. What we do is that, you know, we study the Caucasian population in the United States. And, you know, then we do the measurements of height, head length, you know, feet length, whatever you want to measure, body mass, and weight. And then we draw the curve, and where the curve is settled, we decide, well, if you're in the mean, you are an okay person. If you're above the mean, well, you are tall. If you're below the mean, you are short. So you do an entire population-based statistics. And we thought, you know, maybe we should do the same thing with proteins and then do the calculations of intrinsically disorder. And using these population proteomic-based measurements, we can establish guidelines that they are supported by statistics. And, you know, that's what effectively we did. So what we decided to do was doing a quick literature search and finding which are the most common proteomes that are currently studied in the literature as animal or plant models. And, you know, we selected the 10 most commonly cited. We downloaded the proteins from those proteomes from UniProt, and we used the UniProt reference proteome. We eliminated all those proteins that didn't have full amino acid sequence that were not determined as well. And, you know, we end up leaving just a small group of eligible proteins, which, you know, represents most of the time over 90% of the proteome, because now proteomics is a pretty good science. And, you know, from those, we decide to take all those proteins and systematically use the algorithms that we were given access to do the analysis, which was the other challenge that we had, and estimate the proteome-based statistics. So, and the algorithms that we could find in the literature that authors were willing to openly share with us, there were only 2 algorithms. Surprisingly, we contact most of the people working in the field. There are over 50 algorithms available in the literature. Nobody wanted to share their algorithm. So we only find 3 groups who were willing to share with us. One was the IU-PRED group, because they have it as an open access, and this is a disorder prediction algorithm that actually, what it does is a comparison between globular proteins and those, and it has an index for calculating the energy of globular proteins, and it's an average pairwise interaction of the energies. And then when you deviate from being a globular protein, effectively they consider you a disorder protein. So it's a very empirical definition based on how much your structure, based on the energy and the configuration, depart from the typical globular structure. If you are non-globular, you are considered a disorder. It's not a very objective definition, but it has been widely used and considered in the field. And it tends to have some disadvantages that I'm going to show you very soon when we look at the distributions. Then there are other distributions.
Santiago?
Yes.
It's like 48 out of 50 wouldn't share the algorithm.
Exactly.
And the algorithm isn't, the description's not published?
It's published the description, but they're unwilling to share it. And you know, we're fighting for over a year to actually get all the algorithms, and we couldn't. And in fact, so we had our paper rejected multiple times. So we have tried to publish after the guidelines multiple papers to share disorder atlas. And the reason we cannot publish it is because we cannot use more algorithms. And the reason because we cannot use more algorithms is because nobody is willing to share their algorithm.
So how did you get 55 biochemistry journals to adopt the guidelines?
So those are those are through the commissions. So that's for the enzymology. So if you had an enzyme that is intrinsically disordered, so we have managed to know at least as guidelines for measuring disorder for reporting enzymology data. But it's only for enzymology data. So we haven't managed to adopt these guidelines universally across the protein structure field.
I'm surprised because as far as I know, there's at least 2 more groups. One is David Jones in UCL, who has developed disorder PrED that has full source code. And another group is Yao Qi Zhou in Australia, who developed SPINE. Both of them are downloadable.
Oh yes, and we're currently working on those 2. But you know, the rest again. So the reason we started, so we decided to work first with the physicochemical based algorithms. Then there are the ones that are comparison based with databases. So we are adding those 2 currently. But again, so the numbers of those are available. And you know, I challenge you just to try to find them online or even contact the authors to see if they're willing to share the algorithm. It's only a handful. So now that we're in the open science era, so this the state of the art. So you try to get people give you the algorithms. You know, somehow they want to keep it under the hood. Our experience that people who actually have that work more on the protein structure, they're more willing to share their algorithms and even help you to implement them. While the people who work on the protein informatics, particularly on the disorder area, they tend to be very generous on sharing their algorithms.
So nobody else uses or publishes using their tool?
Well, you can use their tool, but you have to use their websites. And they don't allow you to actually use their website to upload an entire proteome. You can only upload 1 protein at a time or 2 proteins. But they don't let you to actually do a full proteome analysis. So that's where the challenge is. You can use their algorithms.
Sounds like 1765 or something.
Yes. So you can use their algorithms on a web server to run only 1 protein, 2 proteins. And you know, we're trying to run more than once. But of course, then our IP address got blocked. Because of course we want to run thousands of proteins, and in the moment we run 100, then we're blocked permanently for an entire year. So you know that has been one of the challenges that we have been facing. So with this, but anyway, let me tell you. So the other algorithm that we use, because I'm a good friend of the group, so it's the DCMB algorithm, and they have effectively in DCMB, which is funded by the European community, they have 3 algorithms: coils, they have hot loops, and then they have RIMAR 465. And coils is not very effective because it tends to overestimate disorder. And effectively what it does, it assigns residues belonging to one of several secondary type structures, so order or coil loops, depending of certain definitions. I don't want to go through the details because I would like to explain the guidelines and time is running out. Then we have the hot loop that essentially analyzes the calls where they have these C alpha V factors, and effectively it's like measuring the angles in the carbon change, and based on that, on how flexible they are, it would give you a probability that you're a disordered protein. And then finally, this one, there is a neural network trained algorithm that actually is using the XRC coordinates in the protein database. And if you are missing those coordinates, then the neural algorithm would classify those proteins as intrinsically disordered. And we tend to like those using those missing coordinates because they tend to be a good reflection of the lack of crystal structure and therefore a high possibility of disorder.
What is XRC?
So the standard coordinates that you don't find in the X-ray structure on the database.
What does it stand for, XRC?
So, Mike would know that. So it would come to my mind now. So give me just a few minutes and it will come. So I need to remember. So
Cross sections?
it's a cross section of the X-ray. So it's I think X-ray cross-sectional structure. But I will search it very quickly. So if I remember by the end of the talk. Okay, so those are the rooms, and effectively what we end up doing from this sample, we use only hot loops and Raymark 46 because they tend to give less false positives than coils. Coils gives a high positive rate of proteins that are known to be crystallized, but I still consider disorder. And of course, we use the other algorithm that we know that it works and is considered pretty robust, so IUPRED.
In the previous algorithm, how do they calculate the energy? Do they need the structure as in?
No, so they calculate from the amino acid structure. So and then they have a neural algorithm that is based as well from a calculation of the energy based on non-crystal structures that they have. So IUPRED is a neural based approach.
But when you calculate pairwise,
so we can go through the details because we're running out of time, and I have to at 1:15 I have a meeting. So let me just give you an example of how the algorithms work. So in case that you haven't used one. So if you take a protein, let's say, so and I'm taking an enzyme because of course I'm subjectively weak for enzymes. So you take lysozyme and essentially you run the algorithm. So you run the algorithm will give you first each individual residue, it would do a calculation of the protein disorder for that residue, and then you have a threshold to determine whether you are above or below the threshold of what the algorithm considers to be a disorder residue. So if you don't cross a threshold, for example, here you have the blue line in here, and this graph is not perfect, and I apologize for that. So only those that are above the blue line. So if you have the blue curve and the blue line, those are the thresholds that these domains in here, they are considered disorder. If the green line, that's the maximum threshold. If you're below there, you wouldn't consider any of those residues to be disorder, except when you cross the threshold line. And each threshold line for each algorithm is different, depending on how they run their calculations. So what we decide to take each one of the algorithms, then measure that threshold line and see what were the percentage of residues that we will have as disorder. And this is what it would look like if you do the dissemble algorithms, and this is what it would look like if you use the IUPRED. And IUPRED, for example, in this case, the threshold is around 50%. So if you're not on that threshold of 50%, then anything that is below it is considered an ordered residue. So it's a pretty straightforward approach. So what we decided to do then, take those algorithms, and then we didn't trust that the algorithms could, when you run in the whole proteome, they could give you normal distributed PDFs, which probability distributed functions. So what we decided to do a non-parametric analysis, so that we will calculate a unique distribution for each one of the proteins when we were doing our analysis. And then once we have the probability density function, of course, we could calculate mean, median, percentiles, and assess the dispersion of the distribution. And the algorithm was pretty straightforward to run. So what we did, then we took each one of the proteins, took the three algorithms that we wanted to study. So the top one is the IUPRED. The second one is the dissemble hot loops. And then the final one is going to be the dissemble REM. And when you look at this, so this is the percent of disorder you will find in each one of the proteins. And now that you look at the percentage of total proteins that are considered disorder, so the entire disorder of the entire proteome, the disorder of the proteome, so it seems to be on average below 20% of the total proteome is disorder, which is a decent number. So nobody has done those statistics before. We did it, and we did it across proteomes. And we have then, of course, the boxes that determine where the median, the mean, and then the 75 percentile are. We did the same thing with the following thing, so the longest continuous disorder domain, that, as I told you, that's considered the standard measurement for disorder. And if it's about 30 residues, then you would consider a protein that is disorder. Now look at the numbers. So if you do it with IUPRET, so all proteins would be considered disorder in the proteins, because you can see that most of the proteome, and that's the 25 percentile boxes in there. So you can see that all the protein would be effectively disordered if you use IUPRET, because you have more than 30 continuous residues which are disordered. If you use hot loops or use the REM algorithm, so the numbers then they go slightly lower, but still 30 is a number that it seems to be around the mean. And this one in here is slightly low. So this idea of having 30 continuous residues in length to measure disorder is now completely debunked in the moment we have done the statistics. And in fact, if I would be doing the statistics, what I want to make sure is that if I have a highly disordered protein, it should be somewhere around those outside the box, well below the regular percentiles, because that's the point where I would know if a protein is anomalous. Like, so when we say somebody's tall, we don't say that somebody's tall because it's an average person. We say that somebody's tall because on average, 75% of us, we have to look up to see that person and ask how the air is up there. So that's the way, I wanted to do definition. And the other challenge that we have, Mike and I, when we're doing the study, is that this is not actually standardized by the length of the protein. So 30 residues on a protein that is of 50 residues, more than 50% of the protein. What happens if you have a protein that has 1,000 residues? Is that significant? Then we create a new index that actually looks at the longest continuous disorder percentage that is actually based on the length. And on average, we find out that this, when it's essentially standardized, that continuous longest distribution, and then you divide it by the total length of the protein, then we find that the average is well below 20%, which makes sense. So you tend to have a smaller portion of the total protein to be pretty low. And those are again the whole residues. But let me show you the statistics now. So these are the statistics that we find out. And if you start to look, for example, at the longest continuous residues, so this is the number that you have to look for the system. Depending on the algorithm you're employing, if it's IUPRET or if one of the two dissembled algorithms, hot loops or the REM, so you find out, for example, for IUPRET, rarely 30 residues is considered the highly disordered, because this is the average value. And you can see that the average value is 59. So then if you look at, for example, hot loops is 37. If you look at the REM is 21. So it's just there about the mean. So the 30 number. So as contrary to everyone thought, and depending on the organism where you are, for example, if you look at Drosophila tends to have more highly disordered proteins, at least when you use IUPRET, than when you're using, for example, the worm the yeast proteome. So when you look at humans, so humans as well, they look at 70, which is more higher than the yeast. So sorry, mouse. So the human then is 60 in here. So then you have so the mouse in here 70. So the numbers are all dependent from three factors. So depending from first the factor of the protein you're employing. Secondly, the system that you're studying, whether you're using the disorder with one proteome motor or another, depending on the algorithm. And then they're as well going to be dependent on the measurement you're going to be using, whether you're going to be using the protein disorder, the longest continuous length, the longest continuous length standardized by the size of the protein, or with another measurement we define in this paper. So there isn't a unique definition. What you have to use is these overall definitions and then do comparisons. which is then what we decide to do with Protein Atlas. So I have this collaborator in the Brehem Center. So Scott is a really fantastic guy. So he has been studying for a number of years this protein CLEC16. And CLEC16, so the loss of CLEC16 in pancreatic islets leads to impaired glucose homeostasis, and glucose stimulates insulin secretion. CLEC16 seems to be playing an important role in the mitochondrial metabolism. And one thing that Scott discovered is that when he put CLEC16 on one of these motors that calculate protein disorder, he saw, it looks to be a highly disordered protein, and he called it an intrinsically disordered protein. And when he approached my group, he told me, I have an intrinsically disordered protein. It's really exciting, and I think intrinsically disordered is incredibly important. I have my doubts when he was telling me this, because I have seen so many people in the past who, they feel incredibly important because they discovered an intrinsically disordered protein, that I wanted to figure out if it was right. And the way that we did it is that he had this hypothesis that in CLEC16, the intrinsically disordered region is important for the regulation in glucose homeostasis and mitophagy. And we have a couple of guys who were studying this. And in our case, it's not because intrinsically disordered region in the protein is fundamental for the binding. It's fundamental because it's separated in two regions, which are highly structured, and it allows flexibility with those two. And when you mutate intrinsically disordered, then that flexibility disappears to allow the binding. So that's our currently hypothesis. But disorder plays an important role in separating those two regions, where effectively the symphony, the binding, the tridimensional binding needs to occur. And then we run the analysis. So we said, let's just take CLEC16 in the mouse, then look at the mouse proteome, do the comparison on where it stands with respect to the mouse proteome. And when you look at CLEC16, so it doesn't contain a highly unusual number of disordered residues. This is the IU-PREP, this is the hot loops, this is the Riemann 465. These are the distributions of the entire mouse proteome. And if you look where the median, the mean, and they're sitting down, and where CLEC16 is sitting down. So CLEC16, is sitting down there around 30, which is very close to the expected value. It's an average protein when you look at the total number of residues. And it's an average protein not only for IU-PREP, it's an average protein for hot loops, it's an average protein for Riemann 465. And you can look here exactly at the number where it stands. So it stands around 60% of the entire population. So it's slightly more disordered than the average protein, but not a lot. With hot loops, it's well below. And for the Riemann 465, it's slightly above, but it's very close to the average, 65 percentile. I wouldn't say that person is tall if he's on the 65 percentile. Almost average. But what happens is the following thing. So we carry our analysis with the other measurements we did, and what we find out is CLEC16, the disorder is highly organized, because when you look at the longest continuous domain of CLEC16, it stands by itself with respect to the other longest continuous domains in the rest of the proteome. So when you look at the distribution of the longest continuous domains with IU-PREP, the hot loops, and the Riemann 465, these are the distributions. I'm showing there the median and the mean, and look where you have CLEC16. It's in the 89 percentile, so the 90 top percentile of the population. When you look into hot loops, it's on the 90-70 percentile. And when you look into Riemann 465, it's in the 92 percentile. So it's an anomalous protein. In the moment you see that with respect to the population, then you are seeing the really high, tall basketball player, and you have to look up. So there's something unique here with respect to the entire population. And that's what we learned. So that the longest continuous domain is literally important and is unique with respect to the rest of the proteome. And that's what I would call, by our standard definition, an intrinsically disordered domain, because it's well above the mean and on the top 10 percentile of the population. How you can use disorder atlas? So essentially we put this on an algorithm that can do that automatically for you. And then this is a description of what happened with CLEC16 and where the domains are. But what we do is that we put a disorder atlas, which essentially is a web service that allows you to do effectively two things. Do you want to know the statistics about the proteome? So we have the 10 proteins that we did the calculations there, and it will tell you what are the means, what are the standard deviations, how the distributions would look like, what are the bottom 25 percentile, the top 25 percentile, the bottom 10 percentile, the top 10 percentile, so that when you do a prediction, it gives you an idea of where you're standing. But the other thing that it does is that if you have a protein of interest, it could be one that is already on any of the databases, you can call the IU prot number and essentially would run the analysis for you and compare it with the proteome, or you can upload your own sequence and then do the analysis. And I can show you very quickly how it works. I think it was pretty well designed by Mike, and I'm very proud of the work he did. So let's just open here the browser. So if you type disorder atlas together.org, it would bring you to this disorder atlas.med umich.edu, and essentially it would bring you to a panel where you have three options. So let me just. So you can analyze a protein of interest, that is what we have in here. You can search for a specific protein feature, but essentially you can browse the disorder in the proteins. And, let's assume that we want to use the proteins. And it's uploaded there, and then you decide, well, let me just look at the proteome of human. And then when you submit it, it's going to give you all the statistics and the distribution for the human proteome. And effectively you have, the number. So when we did the last accession, so the number of proteins that we have in the database and the ones we use for the statistical assessment, then you can display the proteome description. So it will give you the distribution for each one of the disorder, and it will give you as well the consensus between all the residues that are common between each one of the predictors. It will give you the percentage where they stand. It will give you a table as well with those, and in addition to that, it will give you what is expected value. And that would be, okay, sorry. Okay. So then we have the continuous longest domain, and we have as well the distributions where the percentiles and the number of residues that you require to have. And then we can have as well the balance, that longest continuous residue that is actually balanced by the size of the protein. And we have as well the statistics in there with all the percentiles and tell you where you stand. So if you like to analyze a specific protein, and I think I put the protein sequence of Clex16 in here, so we can analyze a protein of interest. You can do it by the ID, unique prot ID, or you can do it by the sequence. So if you have your protein in here, so let me just do Clex16 in here. So we go back to the browser, log it in, and then you select your organism. And in this case, let's just select the organism where the protein sequence was taken. And then it would give you the analysis. And one thing is that some of these protein disorder algorithms, they give ideal predictions depending on the size of the protein. Some of them they tend to exceed thresholds where the predictions don't become effective, and it will give you a warning message of what would happen if you exceed the number of amino acids. What are the algorithms that you should be using and the measurements that we consider more objective. But so one thing that it would do is that it would give you all the statistics about the protein. So not only the protein length, so the protein where it has been analyzed, but it would give a mean hydropathy. So it would give you this Urbansky atrophy. So the fraction of charge residues, the net charge per residue, the proline contained, and the kappa, which are measurements that people use. It would give you the disorder and order promoting regions, and it would mark those. It would give you the charges where they are. That's for the overall protein. It would show you the propensities of what they look like and where the thresholds are for the protein. It can show you the disorder content as well. So with figures, the distributions, and where you stand. And then it would summarize the statistics as well. And then it will give you the same statistics for the longest continuous domain, showing where you stand, like I showed you before in the presentation. And we have here the consensus. And the interesting thing is that even on the consensus, of course, as well, it gives you high value. So you're in the percentile of 69% on the consensus. And then finally, well, it will give you as well the longest continuous disorder region by the percentage of length. And in this case, if you look at the numbers, I didn't show you those. So you are based on the length, is not that anomalous. So the protein is close to the average values. And then we can give you as well the summary of all the results of what we have been finding out. And the interesting thing is that even you can copy the figures, take them, so it gives you options of exporting the figures in the system. So you can load the image if you want to have a presentation. It's a pretty nice tool. And the challenge that we have is that we want to get it published, but to accept truly as an atlas, even though we call it atlas, we need to increase significantly the number of proteins we have in there. So 10 proteins nowadays is considered like a grain of salt in the number of proteins which are available. And the other thing is that we need to increase as well the number of algorithms that predict disorder. And that's where, even though we don't feel any challenge on downloading non-proteins and doing the analysis, we are finding challenging to actually get the algorithms, installing them in our local servers, and actually running them, because most of them, they are not open access. So with a few of them that you can find in the literature, and some of them we haven't chosen because we don't consider that they're pretty reliable when they come into the predictions, it's difficult to get. Those are considered the most rigorous from the physicochemical point of view, get the authors to agree to give them to us. Well, that's where we stand. So if somebody would like to work on this, so the doors of my lab are open. It's the only omics inclined topic that we have in my laboratory. The other ones are very mathematical inclined. So by informatics students, in my experience, they don't like the rest of the work we tend to do because it's heavily mathematical inclined. But this is something that is more within the lines of what most of the PhD students in the bioinformatics program they do. And well, if you have any questions or comments, I'll be more than glad to answer them.
I'll do first.
How is this different from mobile light and this probe?
Sorry?
So there are already two publicly available databases. One is this probe, one is mobile light.
Oh yes, so different from those that just give you the disorder numbers, and they're not set up in any context. So, and one of the challenges that we have with, for example, with some of these large databases of protein disorder, is that they have even calculated the protein disorder with proteins that their existence is either questionable or they have residues which are not fully defined. And the algorithms can give solutions to that because they have ways to come across them. Well, for example, there is an algorithm, I don't remember which one of these, but it's one of these algorithms that if you have an undefined amino acid, you put X as an amino acid, it would essentially shrink the protein and reduce. If you have 10 amino acids which are undefined, it would shrink it by 10 residues. And then it does the calculation with a protein that actually is effectively different from the original protein. But what you find in the literature, you don't find anything that has been standardized statistically with respect to the whole proteome. Just find pure reporting that hasn't been cleaned up. So that's where I have noticed the difference.
And also, all your data analysis and the statistics are based on prediction, which are not directly derived from experiments and have no experimental verification afterwards.
Yes. Well, this is what happens with most of the protein disorder algorithms. They are just predicted. And again, so, the challenge with protein disorder is an incredibly difficult field to work on because the definition of what a protein disorder is the lack that we have an experiment that proves that it's disorder. It's the lack that it has a protein. So it's a very awkward definition. There is a Science or Nature article that was published a few years ago on how controversial the definition is, because it's a controversial that lacks of physical evidence. You are disordered because, well, we cannot crystallize you. Or you are disordered because we observe multiple configurations. We're not entirely sure if it's the result of flexibility or in reality you're intrinsically disordered. And that's a challenge with this problem itself. But because the definition is widely used, I go to study sessions and people talk about disorder like if something that is physically that you can touch. And the way they know that it's something physically that touches because they use these algorithms for doing the predictions. if we're using the algorithms, I think they should be used in a standardized manner to give you objectively a definition of what it means to be disorder.
If the crystallographer are smart enough to find the right binding pattern to those, a lot of those will settle down, become ordered.
Exactly.
And then if the temperature is lower, they will somehow settle into a low energy state, give up on that high entropy state.
Exactly.
So, okay.
That can happen, although there are some proteins that they have lowered the energy as much as they can.
Yeah.
And on the lowest free energy state, they still observe that only part of the protein can crystallize, and then there are these sections that are very, very flexible.
Are these high entropy regions also mutation prone or selected or conserved?
There are some of them which are conserved. The reason they're conserved, there are people who have multiple hypotheses of evolutionary of why disorder appears.
Yeah.
For example, in plant science, people claim that disorder proteins are incredibly important in the seed phase because essentially they would give volume to protect the embryo while it's developing or is on this fragile state that is not growing. And there are people who actually as well claim that disorder exists because you need to separate it to order regions, and it has that function. So they have exist and they have evolved with potentially having these high entropy states to allow order to exist. But whether that's the case is highly controversial. In the case of the defenders of the theory of protein disorder like Kit Donker and Vladimir Orbiskin, they claim that disorder itself has a function. The flexibility has a function by itself. And they say that their discovery of protein disorder has break down this key and lock mechanism of how protein function actually works.
Therefore, the word native state is actually inaccurate.
Mm-hmm.
These are not native state.
Exactly.
Native state, the key always go with the lock. They're well packed.
Mm-hmm. So. But again, so it's a challenge within the field itself.
Yeah.
And there are biochemists who, if you talk to them about protein disorder, they would walk out and say, look, you have no idea what you're talking about because nobody does. And I don't respect you as a scientist. While others, they take it very seriously. And I think nowadays, most of people are taking it slightly seriously because their algorithms have been proven to be relatively good predictors up to this point.
So, since you're using multiple tools to sort of generate your result, say you got contradictory information from two separate methods, how would you go about resolving that? How would you interpret that result? Which do you trust more to get it right than the other?
Well, in our case, so the guidelines we're developing and we have developed, they're unique for each one of the algorithms. So you have to take it within the algorithm itself. We try to come with this consensus of residues which are common, and that's one way that we have find out to resolve the potential contradictions. But each algorithm would have a unique way to define intrinsically disordered domains of proteins. And what you have to do is trust whether that definition has been set in the algorithm beforehand, it makes sense. And again, because there are more than 50 algorithms, there are some of them that are rarely used. They have been developed by one group, and it's only that group and their friends who actually are using it. While others are slightly more accepted by the public, by the general scientific public. So, and it's difficult to know which one is the right one. And if there are contradictions between them, that's why I think the guidelines exist. Because if you look at our guidelines across the proteomes, somehow there is a common trend. So when you find, and we have discovered that when we analyze a protein, if the protein tends to be disordered with one of the algorithms, the other algorithms tend to predict the same thing. It's not exactly on the same percentile, but it tends to be above the average when we find it out. It's not going to be on the 90 top, on the 10 top percentile of the population, maybe on the 25% top of the population. But it would still, across the disorder algorithms, there tends to be some consistency, at least with the ones that we have been studying so far.
How consistent? So how consistent are different algorithms? Like what is the Matthews correlation coefficient between impaired and DCMB?
No, nobody has studied that, so it's an area that is still open. And again, it's difficult to study because very few people allow you to use an algorithm systematically to do a proteome-based analysis.
Do you have the data? Right?
No, I have the data, but only for 3, and I still feel that, with 3, the analysis shouldn't be, I don't think it's going to be sufficiently solid.
Yeah, but you could at least compare these two where they
Oh, yes, and we have compared them, for example, with the sample algorithms. There tends to be a correlation between their results. They are more closely correlated than the results with IUPREP. And you can see that based on the distribution. So if you look at the distributions that we have run, let me just go back to the presentation. So when you look at the distributions, so you can see that, for example, hot loops always tends to have, like a more normal type distribution, while then the IUPREP and the Remax, they tend to be more similar. So there's a stronger correlation between these two and this. And when you look at, so we have the hot loops and the loops, those two correlate very well between each other. And it's the way they define disorder themselves.
Any last questions? Thank you.
Okay.